Chapter 14 looks at what we call innate immunity or the innate immune response. There are two types of immune responses that our body can mount. One is called the acquired or specific immune response. The other one, which is the one we're going to deal with today, is called innate or non-specific immune response. Okay. So just be aware that these immune responses have different names, and you should be able to recognize them regardless of the name. So innate is also called non-specific. It is called innate because that means we're born with it. So that's the immune system that is protecting us from the time we're born. Versus the other one, which is called acquired, because it will not be active until after we're born, after we start to come in contact with infectious agents. This immune system is also called non-specific, and that refers to the response, which will be of the threat, versus the other type of immune response called a specific, and that one is called a specific because it the response it mounts is tailor-made to the threat. Okay, and then what we have after that are the components of innate immunity, which we're going to talk about. And those are barriers, toll-like receptors, complement immune system cells, cytokines, inflammation, and fever. All right, so we're going to go over through all of these. So the first line of defense are what we call the barriers, and those are the membranes that shield the interior of the body from the external environment. And the two most important barriers will be the skin and mucous membranes, with the skin being the outermost and probably sometimes it's called the first line of defense. Mucous membranes are the membranes that line entrances into the body, like respiratory tract, GI tract, genitourinary tract, etc. All right. So, sensor systems is a series of receptors that detect microbial components. For example, they're going to detect molecules that are unique to bacteria, like peptidoglycan, for example, or lipopolysaccharides. These receptors are found on the surface of most of the cells in the body. And here you have a receptor that detects peptidoglycan. Remember, peptidoglycan is unique to bacteria. Here's another one that detects lipopolysaccharides. That is the component of gram-negative bacteria. Flagellin, again, the protein that makes up the flagella of bacteria. Bacterial nucleotide sequences. When these receptors detect the presence of some of these substances, there will be a chemical change inside the cell that will go into the nucleus, not the nucleus, but again, well, that's a whole cell. Sorry, I was thinking bacteria, into the nucleus and affect gene expression. And the types of proteins that will be synthesized will be those that will be secreted and will alert the immune system as to the presence of infectious agents. So these are non-immune system cells that can detect these bacterial components. In response, they're going to alert molecules that are going to alert the immune system of the presence of infectious agents. Complement proteins are proteins made by the liver. They are immune proteins, and they are found in the blood circulating in an inactive form. The presence of bacteria is going to activate complement, and we'll see what happens here in a minute. Phagocytic cells are cells that can roam around and find and destroy infectious agents, bacteria. Cytokines are chemicals secreted by cells, immune system cells sometimes, often, which are involved in the immune response. And then the overall inflammation, the response of innate immunity to a threat is inflammation. So we're going to go over these components and talk about them a little bit more extensively. Barriers, again, skin is the most important barrier, is the number one line of defense. Mucous membranes, especially those that line digestive tract, genitourinary tract, and respiratory tract, are also protected. The features that make these barriers protected are sometimes physical and sometimes chemical in nature. Chemical immune components of the barriers that are protected will be things like enzymes called lysozymes, which rupture the peptidoglycan of bacteria, peroxidases, which again are going to shut down some bacterial enzymes. Lactoferrins. Lactoferrins are proteins that bind to iron. And iron is a precious metal that a lot of precious element that many cells need for their enzymes to work, and that includes bacteria. So when there is an alert that an infectious agent is in the body, lactoferrins are secreted to sequester iron and prevent iron to be available for bacterial growth. Sweat has antimicrobial components like lysozymes and peroxidases. Fatty acids are also antimicrobial because they are acidic and because they're difficult to metabolize. A low pH is always antibacterial. We know that most bacteria are neutrophils. And the normal flora in our bodies will secrete chemicals that will kill their competition. Those are called bactericidins. So you should know some of the chemical components that act as barriers. Physical barriers will be things like do lactoferrins cause anemia? No, they don't cause anemia. On the contrary, they're going to prevent iron from being eliminated from the body. And yes, iron is a very precious element in the living world because it is very difficult to obtain. So once a system, a cell, in our case, an organism, obtains iron, it is not going to let go of it easily. It will try to retain it. Storing it, especially in the liver, it will store the iron. So no, just because it's being sequestered is not going to cause anemia. It's being sequestered to prevent it from being utilized by bacteria, if anything. So yeah. Actually, bacteria will have their own versions of lactoferrin, which are sometimes even better than ours. So the physical components will be, again, just the washing by urine or saliva of an area that physically removes microorganisms. Ciliated cells, and remember, if you remember from last semester, ciliated cells lined the respiratory tract. Most of the respiratory tract was lined with pseudostratified ciliated columnar epithelium, so that cilia traps microorganisms and sweeps them out of the body. Dryness of the skin, for example, the thickness of the skin is protective. Normal floral competition, mucus, which again is found in a lot of these internal membranes, like the GI tract membranes and respiratory tract membranes. So know the physical and chemical characteristics. Okay. This explains why the skin is considered the number one line of defense. The most difficult barrier to penetrate is the skin. As a matter of fact, unless there is a mechanical injury, there is really no way to go through the skin for an infectious agent to go through to the skin. So it's a formidable barrier. In large part because of the keratin, of the layers and layers of dead cells, keratinized cells, which are going to prevent the entrance of microorganisms. Mucous membranes, again, are typically have mucus or cells with cilia. Some of the antimicrobial substances secreted by skin and mucous membranes will be lysozymes, lactoferrins, defensins. So be prepared to recognize or come up with some of these examples of chemical protections. All right, cells of the immune system. And again, we're talking about the non-specific or innate immune system. White blood cells are, in large part, cells of these innate system. The number of cells during an infection increases whenever we're having a infection. A lot of these cells are white blood cells, leukocytes. We'll talk about those. Okay, so we have two kinds of white blood cells. We have the granulocytes, which are white blood cells that have granules, and they are, for the most part, phagocytic. Then we have large cells, which are phagocytic cells. We have dendritic cells that have some phagocytic activity, and then lymphocytes, which are also white blood cells, and they do not, but however, they're not part of the innate immunity. Lymphocytes. Lymphocytes are in blood. Oh, dendritic cells. Yes, they are. Yes, these are cells that roam around. I did say they were in blood. They're not. These are cells that roam around tissue. So they're found in nearly every tissue. And in some tissues, they have individual names, like Langerhans cells in the skin. So, yeah, they're essentially the same cells, but sometimes they have individual names depending on the place where they're found. Yes, correct. So that's dendritic cells. Langerhans are dendritic cells. Correct. The liver has their own version. I think they're called Cooper cells or Kepler cells, something like that. So they're found all over, and their names may change depending. Yeah, there you go. Kupffer cells. Yep. So they're essentially the same cell. So these are cells that get around. They're all over. As far as the white blood cells, neutrophils are phagocytes, and they're also what we call the first responders. These are the first cells that arrive at an area of infection. They begin to eat up the bacteria, they die, and they create pus. So pus is made by these dead neutrophils. Basophils are not phagocytic cells. These are cells that initiate the inflammatory response. Eosinophils could be phagocytic cells. Typically, they are not. Typically, they are anti-inflammatory cells. So definitely know the basics of neutrophils, what they are, basophils and eosinophils, which are three of the three granulocytic cells, white blood cells. Another white blood cell that is, in this case, it's not granulocyte, is a non-granulocytic cell, are called monocytes. Monocytes are also macrophages. Macrophages. They are eat up other bacteria. They're called wandering monocytes because some of them can leave the blood vessel and begin to roam around in tissue, and then we call them macrophages. So macrophages and monocytes are essentially the same thing. What changes is their location. Dendritic cells, and you'll see from the next picture that dendritic cells are actually relatives of these macrophages. They run around in tissues. Also, they're called scouts because they're looking for things to eat and looking for abnormal cells or tissue. And once they find it, they're going to engulf these abnormal molecules and take them to the adaptive immunity to react to them. So here are the three granulocytic cells. This is the monocyte, which will turn into a macrophage in the car, or in this case, dendritic cell, in the tissue. Sorry, I don't know what I'm thinking, what I'm talking. Macrophages in tissue. Dendritic cells are also tissue cells, whereas monocytes are blood cells, white blood cells. They're both very similar in what they do. The difference would be that dendritic cells do not completely destroy bacteria, whereas the other two do. And then the agranulocytes are not part of innate immunity. Those would be the lymphocytes. So know the cells, know the characteristics of cells. And during the non-specific immune response, there is a chemical communication among cells in order to regulate their function. There are chemokines that attract immune system cells to an area of infection. Tumor necrosis factors are substances that kill tumor cells. Interleukins are secreted by white blood cells, and they're involved in this specific or non-specific immune response. Colony stimulating factors are the chemicals that will determine what kind of blood cell a germ cell will become. But the cytokine we want to spend a little bit of time on is interferons. These are small proteins which are important in viral control, in control of viral infection. And this is the way interferon works. If there is a virus that infects a cell, the response of the cell to the viral infection is to secrete interferon. Interferon binds to receptors of healthy cells and prompts them to transcribe and translate a specific group of proteins called antiviral proteins. So we could have the virally infected cell completely gone, but the interferon which the cell secreted may still be around to affect the non-infected cells. So once interferon is detected by the healthy cell will begin to secrete antiviral proteins. If a virus happens to infect these cells, they will try to destroy the virus, and the cell will try to destroy itself, going to apoptosis or cell death. Okay, so those are the cytokines, and let's pay special attention to the interferons. Toll-like receptors are these sensor systems that alert other parts of the immune system to the presence of an infectious agent. Okay. So this is a picture of toll-like receptors, as we talked about. What you need to know from this picture is the examples of microbial molecules these receptors are going to bind to. So flagellin being one of them. Peptidoglycan is another one. LPS is another one. So be prepared to come out with examples of substances that are detected by toll-like receptors. Okay. There's another set of proteins that are involved in immunity. Those are the complement system. These proteins are made by the liver, just like interferon. Well, interferon is not made by the liver. It's another one. What did we say? Oh yeah, lactoferrins made by the liver. So these are liver proteins. They circulate in the body in an in a inactive form, directly or indirectly, they're going to be activated by microorganisms. So there are three mechanisms to activate complement: the classical pathway, the lectin pathway, or the alternative pathway. The classical pathway is activated by antigen-antibody complexes. So that would be a bacterial cell with antibodies bound to it that will activate the complement system. The lecithin pathway is has to do with receptors on the host cell that are going to be bound by the infectious agent, and that's going to trigger a response, the initiation of the cascade of turning on complement proteins. And then the alternative pathway relies on something called C3b on cell surfaces to when a bacteria binds to the C3b, that is going to activate complement. So C3b is a complement molecule that is bound to bacterial surfaces and that activates the C3b. So what you need to get out of these, directly or indirectly, you need the presence of bacteria to activate complement. Okay, so in the presence of bacteria, complement will be activated. The first complement that begins to be activated in large quantities is C3. And when activated, C3 is split into a C3a and a C3b. Okay. Then the C3b will activate C5, and C5b in turn will activate C6, 7, 8, 9, etc. Okay, so this is what we call a cascade of activation. Most important than all these, though, what you need to know is what happens after complement is activated. So the end result of complement activation, and that's going to be threefold. First, inflammation, then opsonization, and then finally, lysis of the bacterial cell. So, inflammation, complement will destroy. So what marks for destruction may be antibodies, but complement itself will destroy. So these are pretty powerful molecules. They're going to be involved in inflammation, and we'll explain how. Lysis of foreign cells, so the complement itself is going to lyse the cells, and then something called opsonization, which we'll talk about here in just a second. So complement itself could kill a cell by membrane attack complexes, by these method right here. So definitely know the three end results of complement activation. So how is complement involved in inflammation? Two components, two complement. C3A and C5a are involved in inflammation. They're going to trigger changes in the endothelial cells of blood vessels. Then the endothelial cells will begin to separate from each other, creating pores, which are going to make the contents of blood to begin to leak out into tissue. So an increase in vascular permeability associated with inflammation. So in these instances, the blood vessels become leak because of C3a and C3b action, not C3b, C5a and C3a action, and the increased leakiness or vascularity, permeability of the blood vessels are partly one of the signs and symptoms of inflammation. C5a also attracts phagocytic cells to the site of inflammation. So know what happens. How does complement enhance inflammation? Okay, next thing that complement can do to a bacterial cell is opsonization. And opsonization has to do with tagging the bacterial cell for destruction. So we have a bacterial cell here somewhere. Okay, so here's a bacterial cell, and especially the ones that have capsules are very difficult for a phagocytic cell to phagocytize the cell. And the reason why is because the cell, the host cell, the immune cell, does not have receptors for the bacteria. So there's no way for the cell to grab a hold of the bacteria, especially if the bacteria has a capsule. Okay, so here comes opsonization. C3b is the complement molecule that is going to opsonize. It is going to, C3b is going to cover the bacterial cell. That's called opsonization. And the phagocytic cell actually has receptors for C3b. So the phagocytic cell has C3b receptors. So that now the phagocytic cell can actually engulf the bacteria. So here you have the red circles are C3b, and the dark red structures are the receptors in the bacterial cell grabbing a hold of the C3b, with it grabbing a hold of the bacteria and beginning to engulf the bacteria. So another role of C3b in opsonization, another role of C5a in chemotaxis, and C3a in making blood vessels permeable and enhancing inflammation. So that is opsonization. I skipped one here. Okay, the third end result, remember there's three end results of complement activation: inflammation, enhancement of phagocytosis, enhancement of inflammation, enhancing phagocytosis through opsonization, and lysis of foreign cells. So the third one is the one we haven't talked about yet. So the complement proteins that are involved in these, in lysis of foreign cells, are called MACs, which stand for membrane attack complexes. And these are proteins C5b, 6, C7, C8. And these molecules are going to insert themselves in cell membranes. And actually, let me rephrase that. It's going to insert itself into cell walls and cause the cell to become leaky. It is important to know that these complexes, these proteins, insert themselves into a cell wall that has the same component as cell membranes. So notice that this is a phospholipid cell wall. And this is what the cell would look like under the microscope, with all the little pores created by the complement proteins. Yes, this is different than making a cell competent. Good thinking. Yes, it is. When you make a cell competent, that is being done by electroporation, electricity shock, or exposure to salts. This is different in that these are proteins that insert themselves into the cell, into the cell wall, the phospholipid or lipid cell wall, and creates pores. So yeah, it is not the same. I was gonna say one more thing. Okay. So yeah, I remember now what I was gonna say. Okay, so these complement proteins insert themselves into phospholipid membranes. Okay, what that means is that MAGs are not going to be as effective against gram-positives because gram-positives have a peptidoglycan cell wall, not a fat cell wall. Like gram-negatives have LPS, lipopolysaccharides. So they have a lipid cell wall, which can be attacked by MAGs. So MAGs are more effective against gram-positives than gram-negatives. The other way around. MAGs are more effective against gram-negatives than gram-positives. I'm sorry about that. Because gram-negatives have a fatty cell wall. And that's where MAGs attach or insert themselves into fat cell walls. Okay. Okay. Chemotaxis and phagocytosis are very much linked in that chemicals secreted by injured cells or complement molecules, C5a, act as chemotactic substances. These are substances that attract to the area of infection immune system cells. And by attracting phagocytic cells to the area where the microbes are, now phagocytic cells can eat up this bacteria, especially if it has been opsonized already by C3b. So phagocytosis is very much enhanced by complement. C3b complement opsonizes bacterial cells and makes phagocytosis more likely. C5a attract to the area of infection phagocytic cells. C3a, it's making the blood vessels porous so that these cells can leave. Now, what happens after the cell is phagocytized? The cell is going to be brought into the phagocytic cell, the bacterial cell is going to be brought into the phagocytic cell in a little vesicle called a phagosome. Phagosome is actually a nice place to be for a bacteria because it's hidden away from the immune system. However, phagosomes are going to join with little killer sacs called lysosomes. These are also called digestive sacs. The two come together into what's called a phagolysosome. And the digestive enzymes of lysosomes begin to digest the bacteria. Bacteria is digested and then released by exocytosis. So you should have an idea of what happens when a bacteria is phagocytized. Know the difference between a phagosome versus a phagolysosome, which is the fusion of a phagosome and a lysosome. Know the types of substances that lysosomes have that will kill the bacteria. Things like low pH, oxygen radicals, digestive enzymes, defensins, lactoferrins that will take away iron. Okay, so these are pretty destructive little sacs, these lysozymes. All right, so you have an idea of what happens during phagocytosis. And one thing to keep in mind, this material comes back on chapter 16. So we're looking at these interactions between the immune system and bacteria from the immune system point of view. And we're looking at how our immune system attacks bacteria. In chapter 16, we're going to reverse that, and we're going to look at what bacteria does to defend themselves from these attacks by the immune system. Okay, so as you're looking at these, you can think, okay, what could bacteria do to prevent phagocytosis? It would have to try to destroy C3b. It could destroy C5a, and that alone will prevent the immune system cells from arriving, period. It could try to prevent the phagolysosome appearance, prevent lysosome-phagosome fusion, for example. Okay. So we're going to come back to all these and see what bacteria do to try to protect themselves or defend themselves from these kinds of from our immune system. Okay. And all of these components come together in the main response of the non-specific immune system, which is inflammation. Inflammation is dependent on inflammatory mediators, meaning on chemicals that initiate the events of inflammation. These could be chemicals secreted by cells like macrophages, but typically it starts with chemicals secreted by damaged tissue. Do you know the signs and symptoms of inflammation and what causes those signs and symptoms? So heat is due to the increased circulation. Pain is due to the fluids escaping from circulation, and the excess fluids are pressing on nerve endings, causing pain. Redness is also due to the increased circulation. Swelling is due to the increased blood vessel permeability that will allow for the escape of fluid from the blood vessel. Sometimes loss of function is mentioned as the fifth sign. So here's an inflammatory event. You have some type of injury on the skin that's going to cause C5a and C3a to dilate blood vessels and make them permeable. And inside blood vessels, what's going to happen is that a couple of things. Remember that blood vessels, capillaries, are made of endothelial cells. So these little flat cells, especially if they've been injured, they're going to begin to put out these receptors that are going to trap white blood cells. So these are the cells that are close to the area of injury that put out these receptors. So the white blood cells get trapped by these receptors and they're going to flatten. And after they flattened, this flattening of white blood cells to the walls of a blood vessel around the area of inflammation is called margination. Next, the cells begin to put out extensions of their cytoplasm, which are going to be extended into the pores created by C3a. These are called diapedeses, these extensions of the cytoplasm that look like little feet with which the cell leaves the blood vessel through the pores. There is a depolarization of the cell membrane when they're injured, which will trigger the release of inflammatory chemicals. So yes, that's all part of this entire positive feedback cascade. Is that there is the injured cell is depolarized, influx of either calcium or sodium depolarizes the cell, and in response, the cell begins to secrete inflammatory chemicals. That's kind of like a signal that something has gone wrong. This is the C5a attracting phagocytes at the area of inflammation. Okay. Factors that initiate inflammation. So we talked about white blood cells putting out inflammatory chemicals. We talked about endothelial cells, injured cells, put inflammatory chemicals. Microbial products can do the same thing also. Microbial products could also initiate inflammatory, put out inflammatory chemicals. Okay. Also remember that the microbe themselves can trigger complement activation. Okay. So again, go over the signs and symptoms of inflammation. Outcome of inflammation is to limit tissue damage and restore function. However, inflammation itself can cause considerably damage, especially if it becomes chronic, either because it's a tremendous acute inflammatory response that can be extremely difficult to control, or it may become a long-term inflammatory response, which becomes chronic. Osteoarthritis is wear and tear. Rheumatoid arthritis, on the other hand, that is definitely a chronic immune response. So you may have some inflammation going on in osteoarthritis just because of the wear and tear of the cartilage of the joints, but an immune response that is causing a constant, very, very severe immune response will be something like rheumatoid arthritis, for example. And there are some bacterial components that cause a long-term inflammation also. Encephalitis, for example. Well, that would be viruses, not bacteria, but still, that would be an inflammatory response. I'm thinking of rheumatoid arthritis because that's rheumatic fever. Those are all long-term chronic inflammatory responses. I mean, even Lyme's disease ends up being an inflammatory response. Okay. All right. The last component of this nonspecific immunity is fever. And fever is caused by substances, fever-causing substances called pyrogens. The purpose of fever is to increase the body temperature above the maximum growth temperature of the microbe. So if the microbe grows at its best at about 37 degrees, we can increase temperatures to 40, 41, 42 degrees centigrade. And hopefully that will slow down the microbe or right out like just kill it. Okay. Another advantage of fever, especially a mild fever, is that activates and speeds up our enzymes, which with that it speeds up our defenses. Pyrogens are substances that produce fever. We can have endogenous pyrogens, which are made in our own body, things like interleukins secreted by immune system, which are fever causing. Or we can have exogenous pyrogens, which are bacterial products that cause the fever. Things like the capsule of a bacteria may be pyrogenic. Okay. So, any questions over these? Like I said, this is a very quick chapter, not terribly hard. All right. So I would like to go a little bit, this is the wrong one, over chapter 15. There we go. Maybe goes through some of the first PowerPoints, and then we can probably go ahead and stop there. And next week we can wrap up chapter 15 and the little bit of 18 that we covered. So chapter 15 looks at the other type of immunity, which is called adaptive or specific immunity. It's also called acquired. So it's adaptive, specific, or acquired. It's called specific because the response is tailor-made for a specific threat called an antigen. It develops after birth. More importantly, it has a memory. It has an immunological memory. What that means is that the first response to an antigen is not as severe as the second response. The second response is quicker and enhanced because it remembers the first response. So the second time is magnified. The response is magnified. So this is unique to adaptive immunity. Yeah, so the first response will be the first encounter with the pathogen. The second response will be what it will be enhance because of the first response. The first response may take as long as 2 weeks to develop. The second response takes hours, maybe 72 hours. It is fairly immediate and enhanced manyfold. Those are the basis for vaccination. So what the vaccine attempts to do is to give us the first response, so that if we ever encounter the bacteria in our daily living, then now the secondary fast response and enhanced response will kick in and destroy the microbe. Okay. Adaptive immunity, in turn, has two components. So we have immune system has two components: the specific immunity and non-specific immunity. The adaptive immunity versus the innate immunity. So adaptive, innate, acquire is all one type, and non-specific is the other type. The adaptive immune response has, again, in turn, two components, two divisions. Humoral immunity is the branch that eliminates extracellular pathogens that are in the body, typically circulating in lymph or blood. The cells of humoral immunity are called B lymphocytes or B cells, and this type of immunity develops in bone marrow. On the other hand, cellular immunity is going to go after cells that are infected or cells that are abnormal. So they're going to eliminate intracellular pathogens. The cells involved in cellular immunity are the T lymphocytes or T cells, which are going to be created in the thymus. So humoral immunity is developed in the B cells develop in the bone marrow. Cellular immunity, the T cells involved in cellular immunity, they developed in the bone marrow, but then they mature in the thymus. So all cells, all blood cells come from the bone marrow, and all immune system cells come from the bone marrow. B cells stay in the bone marrow and mature in the bone marrow, whereas T cells are taken out of the bone marrow to the thymus, where they're going to mature. Okay. Humoral immunity depends on lymphocytes called B cells. When these B cells, and B cells, all of them, each B cell has a receptor which will be specific to an antigen. And an antigen is essentially a molecule that can bind to a B cell. So if a B cell happens to, or an antigen happens to be in the body, and that antigen finds the B cell that it could attach to, then this B cell that has been attached to by the antigen will begin to differentiate into a different kind of cell called the plasma cell. And plasma cells are the ones that actually secrete antibodies. Some of these plasma cells are going to divide and remain differentiated, because they're going to be the ones that will remember the first response. They become the memory cells. Okay, so this is essentially what happens during humoral immunity. So this is the B cell, and so it happens that a antigen is attached to the B cell. Now, notice that for the B cell to be activated, it first needs to be activated itself by a T helper cell. So this is a T lymphocyte, and it has receptors for the specific antigen. So when the T helper cell encounters this antigen, it becomes activated and secretes interleukins that will activate the B cell into differentiated into plasma cell. So if the T helper cell is not there, the B plasma cell is going to is not going to respond to an antigen. So we need the T helper cells to present the antigen to the B cells. All right. Oh, and also remember, some of these cells will become memory cells. Some of the plasma cells that are made will become memory cells. Plasma cells will be the cells that are actually going to secrete antibodies. So humoral immunity is the immunity that secretes antibodies. Cellular immunity depends on T cells, especially the T cells known as cytotoxic T cells. These are the ones that are going to recognize and kill abnormal cells. Cytotoxic T cells, however, just like the B cells on humoral immunity, depend on T helper cells to activate them. So T helper cells activate cytotoxic T cells and B cells. Okay, so here's the, let's see, here is the cytotoxic T cell, which has been activated by the T helper cell that has already seen the antigen. So the T helper cell has already seen the antigen, and that allows it to begin to secrete interleukins that will activate a cytotoxic T cell that is bound to antigens. So notice that the T helper cell is needed for the activation of the cytotoxic T cells and the B cells. Okay. So it's a good place to stop chapter 15, and we'll wrap it up next time together with the last little portion of vaccines, chapter 18. And so after next Wednesday, no, we meet on Thursdays. After next Thursday, we should be ready for test number 4. No, test number 5. Sorry, test number 5. Okay, any questions?
Will you go over the labs that you mentioned on Monday, this coming Monday?
Okay, I'm sorry. What about the labs? What was your question?
Okay, I know lab 10 was the official last lab report, but I know you mentioned the other labs Monday, so will you go over those this coming Monday?
Yes, we'll cover lab report 11, which is very short, and then we'll go ahead and cover I'll have to think which one would make more sense to cover first. It may be medical microbiology lab to cover first, and then I'll cover the other one. They're both very similar, but they deal with different types of bacteria. So yes, that's exactly what we'll do on Monday.
Okay, thank you.
Oh, you're welcome. Any other questions? Okay. So, if you're gonna have any questions between now and Monday, just shoot me an email. And keep working on these chapters. That way, as soon as we are finished covering them, we can go ahead and take the test and turn to the last set of chapters that we have left. So we're nearing the end here, which is nice.